So today's question paper is about previous year question paper of PG entrance 2024. So now the question is, the specimen used by the author is the nomenclature type is called. See, there are several. It is in the taxonomy. It is called as typification. The herbarium specimen, when an author, when he find a new species, okay, he preserve that plant species in the form of a herbarium specimen. Okay, so that specimen, what he says, he described that specimen, he publishes one paper on publication. Okay, in the form of the publication, he describes the morphological features of that plant species, which is available in the form of the herbarium specimen. Okay, now the one who describes, one who found this species is called as an author. Okay, so the author uses a specimen, the specimen used by the author in the nomenclature type. What kind of nomenclature type? There are different kinds of nomenclature types here: lectotype, neotype, holotype, isotype. So these are all you study in the typification, in the taxonomy. Okay, so the answer is holotype. Okay, the specimen used by the author. Okay, the specimen used by the author is called as holotype. And isotype, because that author or the scientist, he collects more than one specimens from that site, from that geographical area. He collects not only a single one, but he collects more than 2, 3, 4, like that, some specimens. And the specimen used by the author in the publication to describe that plant is called as holotype. And other than that specimen, there are some other specimens of the same species. Okay, so here, what is isotype? The duplicate of the holotype is called as isotype. Duplicate to the original one, the original specimen used by the author to describe that plant is called as holotype, but the duplicate, because we maintain some extra copies, duplicate, triplicate, like that, original, duplicate. The duplicate of the holotype is called as isotype. And lectotype. What is lectotype? Other than the holotype, isotype, the next specimen. That means if we don't find in the future, because we have so many specimens in the Royal Botanical Gardens, which were collected in the 1900, in the 1857, 1860, like that, 100 years back also. So sometimes what happens, they may be lost, like holotype or isotype. If they are not available, another specimen collected by the author, that is called as lectotype. And apart from holotype, isotype, and lectotype, if all of them are lost, that means he collects 3 specimens. The original specimen is holotype, duplicate is isotype, and one more remaining one is lectotype. If all these are lost in any case, then what happens? The same species is collected again from the same geographical area where that author initially collected. One more specimen will be collected from the same geographical area, that is called as neotype. Okay, so this is called as typification. And next one is the rhizoids in Markhamshire are. In Markhamshire, So in Markamsia, yeah, you see the diagram of the Markamsia here. This is the dorsal side. The upper part is called as dorsal side and lower part is called as ventral side of the Markamsia thallus. In the lower side, we see two different structures. The unicellular structures are called as rhizoids and the multicellular structures are called as scale leaves. Scale leaves, they protect, okay? They protect for the main purpose of the scale leaves is for the protection of the thallus, for the birds, rhizoids. So here the main function of the rhizoids is to absorb the water and minerals from the soil. The lower part of the thallus is called as ventral part and it has both the scale leaves and rhizoids. So how the rhizoids are? So you see the rhizoids, these are unicellular but they are pegged inside. Okay, pegged structures. Okay, so that is the main feature of the rhizoids in the Markamsia. So you see this one, the rhizoids in Markamsia are, they give the options smooth, multicellular. They are not multicellular and okay, and tuberculate, multicellular. They are not multicellular. Unicellular and smooth. They are unicellular but not smooth. Smooth means there is one more character, tuberculate. That means budipel. Budipel untai tuberculate. Inside there are tubers like structures. So it is called tuberculate. So unicellular, smooth, and tuberculate. That is about the rhizoids in Markamsia. Next one is pseudo-elaters are presenting. So here, if you see the elaters, where we find the elaters, just I will tell you. In case of the bryophytes, the sporophyte consists of the foot, seta, and capsule. Okay, and in case of the Markamsia, in the capsule, other than the spores, elaters are also formed. The main function of the elaters is dispersal of the spores. Okay, when the dehiscence of the capsule occurs, when the dehiscence occurs, what happens? The spores are released at once, and these elaters are hygroscopic and just like a blast. Okay, when the dehiscence occurs, all the spores are dispersed with the help of the elaters. So the elaters which are present in the Markamsia capsule are called, they are the true elaters. Okay, they are called elaters. But next to Markamsia, there is Anthoceras. Okay, that is also bryophyte. They are also Anthoceras also called hornwort. Okay, Markamsia is called liverwort. Okay, and Anthoceras is also called as, common name is hornwort. In the structure of the Anthoceras capsule, there are the elaters like structures. They are not considered as the true elaters. They are called as pseudo elaters because in the elaters of the Markamsia, there are spiral thickenings. Those spiral thickenings are absent in the elaters of the Anthoceras. Okay, so they are called as pseudo elaters. Okay, they are also useful in the dispersal of the spores. Okay, so Anthoceras is the right answer. Once we'll see this. Yeah, you see the diagram here. This is in case of the Markamsia. Along with the spores, there are structures, filamentous structures with the spirally coiled thickenings are there. So these are called as elaters of Markamsia. But in case of the Anthoceras, in the next diagram, along with the spores, these are the filamentous structures but without spiral thickenings. They are called as pseudoelaters. Okay. So next, who studied the epidemiology of wheat rusts in North India? In North India, wheat is cultivated. Okay, compared to the South India, in North India, wheat is cultivated in large scale. And there is one famous disease that is called as wheat rust of wheat. We know about the Puccinia graminis. It causes a wheat rust. We already discussed about the different kinds of the spores which are present in the life cycle of the Puccinia, okay, which is the pathogen causes the rust disease in wheat. And who studied? One scientist is famous for his study on the rust disease. So here given V.V. Chenulu, K.C. Mehta, P. Ramachar, Y.L. Nine. So the right answer is K.C. Mehta. So he is very famous for his studies on the wheat rust. Okay. So you should remember the K.C. Mehta. Parmelia is a. So what is Parmelia? So here you see in the lichens, there are different kinds of the lichens. For example, crustose lichens, which are very thin. They bind to the rocks. Okay, very strong binding is there. We can't remove them. They are very thin and they bind to the rocks. They are called as crustose lichens. So we also studied them in the xeroxere or lithoxere. That means they are also called as pioneers of the lithoxere. Pioneers. That means they start their life on the rocks, bare rocks. They can grow because lichens contain both the algae and fungi. Both the algal partner, fungal part, both are there, and fungal partners are able to absorb the moisture from the atmosphere and provide it to the algal partner for the photosynthesis. So, and foliose lichens are here, Cora, Parmia. Why they are called folio? Because they are leaf-like. They are the leaf-like lichens, appear like leaves. And here one more example is Parmelia. So Parmelia is an example for the foliose lichens. And one more type of the lichens here, fruticose lichens. They are either filamentous structures. Okay, so these structures are called as shrub-like structure. Shrub-like, they appear like shrubs or filamentous. They are called as fruticose lichens. Example, Usnea and Cladonia. Okay, so crustose lichens form flat, thin, crusty layers. Examples are Graphis scripta. It is also called script lichen, and Lecanora. These two are the examples for crustose lichens, which are very thin. Okay, and foliose lichens. Already we discussed broad, flat, and leaf-like structures. Foliose means leaf-like structures. Examples are Parmelia. The common name is shield lichen. Okay, you have to remember common names also. Parmelia is also called as shield lichen, and Xanthoria, it is called as sunburst lichen. These two are example for foliose lichen. And fruticose lichens. These are three-dimensional and highly branched and shrubby. They are filamentous. Examples are Usnea. It is also called as old man's beard. Usnea, the common name of the Usnea is, it is very famous, old man's beard. And one more important one is Cladonia. It is called as reindeer moss or reindeer lichen. It is also very important. Cladonia. In several areas, this Cladonia, they use it as a feed. Okay, to feed the cattle, and they also feed for they eat them. Okay, cladonia. It is also called as reindeer moss or reindeer lichen, and usnea is also called as old man's beard. So you have to remember these words. Next question is, in Cercospora, conidia develop on. Cercospora belongs to the fungi, and it specifically belongs to the Deuteromycotina. Deuteromycotina, it is also called as an artificial fungi. Or it is also called as fungi imperfecti or imperfect fungi, because there is no sexual reproduction in case of the Deuteromycotina. Okay, among the fungi where we find the sexual reproduction and asexual, only asexual reproduction, they are separated. All the fungi are separated, those without sexual reproduction. Okay, so in Cercospora we don't find sexual reproduction, and we find the asexual reproduction. During the asexual reproduction, conidia are developed. Okay, so this Cercospora is also important. It is a pathogen which causes ticka disease. Okay, it causes ticka disease in the groundnut. There are two species in Cercospora genus, Cercospora arachidicola and Cercospora personata. These two are the species. Now the question is, from where conidia develop in Cercospora? You once again you see this. So this is the ticka disease in groundnut, okay, which is caused by Cercospora. There are two species, Cercospora personata, Cercospora arachidicola. You see the structure here, acervulus, it is also called as. Here you see the from where these conidiophores are produced, from the filamentous structure at the base is called as stroma. Stroma is an interwoven hyphae. The fungal filaments are also called as hyphae. Okay, mycelium and hyphae, these are the terms used with respect to fungi. Okay, so the right answer is stroma. In Cercospora, conidia develop on stroma. That is the right answer. Next, in albugo, the diploid zygote is known as albugo. Again, there are it also belongs to fungi, and albugo it belongs to Phycomycotina or Phycomycetes class, Phycomycetes subdivision Phycomycotina. Okay, there are Phycomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. These are the 5 important subdivisions. If we call it mycetes like Phycomycetes, it is class. Okay, so albugo, it is also called as cystopus, so which causes white rust disease. Okay, white rust disease in Amaranthus, and also in the Brassicaceae members, albugo causes white rust disease. So here what they are asking, the diploid zygote after the reproduction, after the sexual reproduction, the diploid zygote is produced. What is the name of that? Also, it is also known as, the diploid zygote is also known as oospore, zygospore, ascospore, basidiospore. In the fungi, there are different varieties of the spores. Okay, in the Phycomycetes, these diploid zygotes are called as oospores. Okay, albugo, it belongs to Phycomycetes. The diploid zygote of the albugo is called as oospore. And in case of the mucor, for example, in the Zygomycotina or Zygomycetes, the diploid zygote produced is called as zygospore. So that class is also called as Zygomycetes, subdivision Zygomycotina. And another one is ascospore. So ascospores are produced in the ascomycetes. There is a important structure called as ascus. The plural form is asic. Okay, from the ascus, spores are produced. Those spores are called as ascospores in ascomycetes. For example, penicillium belongs to ascomycetes. Yeast also belongs to ascomycetes. And basidiospores, we already discussed in case of the Puccinia, which belongs to Basidiomycetes or Basidiomycotina. There we find different kinds of the spores, iridospores, steltospores. But important one here is basidiospore. It is the characteristic feature of the Basidiomycotina or Basidiomycetes. So now the question is about the albugo, which belongs to Phycomycetes. So the diploid zygote is called as oospore. And there is a difference between the oospore and zygospore. What is the main difference? During the reproduction, if the male gamete and female gamete both are of same size, more or less, also called isogamy. If both the gametes, male and female gametes, are similar in size, in morphology, then the diploid zygote produced in that fertilization is called as oospore. But in case of the zygospore, the male gamete is smaller than the female gamete. Often the female gamete is called as X. In some of the species we find this. So this is called as zygospore. Okay. So you have to remember, in case of the albugo, oospore, in case of the mucor, that is called as zygospore. In case of the penicillium, it is called as ascospore. In case of the Puccinia, it is called as basidiospore. And heritable change induced in the base sequence of a DNA molecule is referred as. So we know Hugo de Vries, he identified mutations. Okay. So mutations are very important because they are inheritable or heritable. That means they are passed on. Once mutation occurs, that mutations will be passed on to the next generation. That's what they are asking. So what is that called as? Mutation. That process is called as heritable change induced in the base sequence of a DNA molecule is referred as mutation. Mutant means the organism which undergone mutation is called as mutant variety. That is the mutant variety. Mutagen, what is mutagen? The factor which causes the mutation. It might be UV radiation, any factor which causes mutation is called as mutagen. And the process of formation of the mutation is called as mutagenesis. Here they asked about that heritable change. What is that heritable change? Is called as mutation. That is the right answer. Next, in which type of mutations thymine dimers are formed? Thymine, you know, in the DNA, there are adenine, guanine, thymine, cytosine. These are the four bases are, nitrogen bases are present. But in one type of the mutation, the two thymines, if the thymines are present next to each other, they are linked and they form a dimer. It is also one type of the mutation. So which type of the mutation? What is the reason for this type of the thymine dimer formation is X-rays, gamma rays, UV rays, beta rays. UV rays, okay? UV rays is the right answer. Here you see in the DNA, double-stranded DNA, here you see the thymines next to each other in a sequence. And when these DNAs are exposed to the UV radiation, then two dimers are linked by the double hydrogen bond and they form a dimer called as thymine dimer due to the UV radiation. So that is the right answer. Thymine dimers are formed due to exposure to UV rays. Okay, next. what is the fruiting body in penicillium? So in penicillium, after the sexual reproduction, one fruiting body is formed. What is that? That is called as cleistothecium. Okay, there are different types of the fruit bodies: cleistothecium, perithecium, pycnidium, apothecium. Cleistothecium is a closed structure. Okay, and perithecium is it's an open structure. You see here. You see, in case of the penicillium, during the sexual reproduction, you see, the male and female filaments are there. Okay, so here female branch is there and male branch is this male branch. It coils around the male branch is called as antheridium, and female branch is called as ascogonium. And this male branch, it coils, it touches the ascogonium, and after that fusion of the male and female gametes, female nuclei occurs, and they form the pairs. Nuclei are formed pairs, plus and minus, and after the zygote formation and ascus, the structure which is formed from the ascogonium, these structures formed are called as ascus. From the ascogonium, the structures which are formed are called as ascus. Several asci are produced. Okay, and all these ascus filaments, ascus and normal filaments, all the hyphae filaments, they form a structure, closed structure, surrounded by the sterile filaments or sterile hyphae, and inside we find several ascus or asci with the ascospores. In the ascus we find the ascospores. So they are all present within the closed structure cleistothecium, which is formed in case of the penicillium sexual reproduction. Okay, that is the right answer is what is the fruiting body in penicillium is cleistothecium. Okay, next. Budding is the mode of reproduction in where we find the budding. It is very important with respect to Saccharomyces. It is also called as yeast. In the yeast, we find the process called as budding. Okay, buds are formed. Saccharomyces or yeast. Which species of Cara is endemic to India? Cara baltica, Cara nuda, Cara stelligera, Cara valici. So here the right answer is Cara nuda. In India, this is endemic. Endemic means what is the endemic? It grows in India only, here only, not in other countries. Okay. For example, red sanders. It is also endemic. It is endemic to some area only. It is found only in the Rayalaseema area. Okay. In some areas of Rayalaseema and some areas of Karnataka and Tamil Nadu, we find this. So these are called as endemic. So Cara nuda is endemic to India, and vegetative reproduction in Cara occurs by. So Cara, it is a green algae, belongs to chlorophyceae. Okay, and we find globules, nucleus, but these are the reproductive structures. Okay, globule and nucleus are the male and female gametophyte. Okay, and here amylum stars is the right answer. With the help of the amylum stars, you see here, this is the amylum star, which is characteristic to the Cara. If you see the fragmentation, that means fragments of the algae are separated and they grow as a full grown alga. But that is present in several cases of the algae. But amylum stars is present particularly in case of the Cara for the vegetative reproduction. They store the food material in the form of the amylum. useful for the growing birds. That is called as vegetative reproduction. Okay, next. So this is all about the 2024 paper. Okay, PG, M.Sc, Botany entrance question paper. That is. So next we'll move to another paper, that is 2023 M.Sc Botany entrance question paper. So here you see the question is Abulmoscus esculentus belongs to the family. To which family? Abulmoscus, it is also called as lady's finger, bendakai. Okay, it belongs to the family Malvaceae. So you should remember some of the plants. And Morels and Agaricus have edible fruiting bodies. They belong to their respective classes. So these two are used, okay, as a food material. ఓకే వీటిని ఏమంటారంటే పుట్టగొడుగులు అని కూడా అంటారు తెలుగులో. ఓకే, సో ఇక్కడ మోరెల్స్ బిలాంగ్స్ టు, మోరెల్స్ ఆల్సో ఎడిబుల్. These are also called mushrooms. Okay, mushrooms belong to various classes. Morels belong to Ascomystis, and Agaricus bisporus, okay, we also call as button mushroom, Agaricus bisporus. And Morels, the botanical name or scientific name Morchella, the common name is Morels. And Agaricus, it belongs to Basidiomystis. So that is the right answer. Morels belong to Ascomystis and Agaricus belongs to Basidiomystis. Next, which one of the following statement is not true? So they are asking out of the 4, one question is wrong. Remaining 3 are correct. So we have to find out the wrong statement. Hexane of pollen grains is made up of sporopollen. It is right, because sporopollen it can withstand aging. Okay, so it won't degrade. Because of this sporopollen, pollens, they are available, even belong to several previous eras. The paleopaleontology, the study is called as paleopaleontology. That means study of the pollen belong to long back, okay, in other eras also. So we can find them in the several earth layers, those pollen grains. We find them as the shells. Okay, so that is one. Next, that is the right one, so we should not choose that. Pollen grains of many species cause severe allergies. That is also right. They cause hay fever, like that. Stored pollen in liquid nitrogen can be used in the crop breeding program. That is also called right. Okay, cryopreservation, that is called as cryopreservation, preservation of the pollen in the liquid nitrogen, and later those pollen can be used for crop breeding experiments. That is also true. And tapetum helps in the dehiscence of anther. That is wrong. For the dehiscence of the anther, stomium is important. Okay, that is present in the epidermis of the anther wall. They are present. They are the enlarged, thin-walled cell structures called as stomia. Okay, the dehiscence, the breakdown of the cell walls of the stomium occurs, which allows the release of the pollen grains from the anther. This process is called as dehiscence. Next, the genetic material in TMV. So TMV, tobacco mosaic virus, is a plant virus. You know, there are different kinds of the virus. Phytophages, plant viruses. Zoophages, animal virus. Bacteriophages, like that. But what is the genetic material of the plant viruses? Usually that is the single strand RNA. So in TMV, we know Stanley, he crystallized the TMV. That is very important. Stanley crystallized the TMV for the first time and he got the Nobel Prize. And the genetic material is present in the TMV is single strand RNA. But there is one exception also. There is one plant virus called as cauliflower mosaic virus, CAMV. It has double strand DNA as a genetic. So you should remember that cauliflower mosaic virus is a plant virus, but it contains double-stranded DNA as a genetic material. That you should remember. And there are some viruses with the double-stranded RNA. There are some plant viruses with the double-stranded RNA also, called as Rio virus. Rio virus contains double-strand RNA. Usually RNA in a single-stranded form, but here in the Rio virus we find double-strand RNA.